Hi everyone, welcome to all. This is Vidyam Academy's most expected question series for medical NEET examination. Today's topic is 12th Biology chapter number 5, Molecular Basis of Inheritance. And into this subtopic is Human Genome Project, DNA Fingerprinting. And now it's being presented by Rajiv Patel. Once again, let's welcome to all. Let's get started. The first question is, the Human Genome Project was one of the largest collaborative scientific projects ever undertaken. Which option correctly states when it was launched, when it was completed, and its primary goal? Option A, launched in 1990, completed 2003. Primary goal was sequence all 3.3 billion base pairs of the human genome and identify all 20,000 to 25,000 genes. Option B, launched in 1975, completed in 1990. Goal was to sequence only the protein coding genes of the human genome. Option C, launched 2000, completed 2010. Goal was to sequence to the entire genome of all mammals simultaneously. Option D, launched 1990, completed 1998. Goal was to map only the chromosomal locations of human disease genes. Would you guess the right answer for this question? So here the options A, B, C, D. This is the question which directly related to Human Genome Project's goals and year of completion. Let's check whether your guess is right or wrong. The right answer, option A. Let's see why and how. Go to the detailed explanation. The Human Genome Project was formally launched in October 1990 as an international collaboration involving research centers in the USA, UK, France, Germany, Japan, and India. CCMB Hyderabad was India's contribution. The first one. The primary goals were to determine the complete nucleotide sequence of all 24 human chromosomes, 22 autosomes plus X plus Y. So totaling approximately 3.3 billion base pairs, and to identify and map all human genes. A working draft covering 90% of the genome was published in Nature in 2001, and the high quality complete sequence was declared finished in April 2003. 50 years after Watson and Crick's double helix discovery, the project revealed approximately 20,000 to 25,000 protein coding genes. For fewer than the 1 lakh that scientists had initially predicted and discovered, the coding sequence comprised only about 1.5% of the genome. That's what option A, launched in 1990, completed in 2003. Primary goal was to sequence to all 3.3 billion base pairs of the human genome and identify all 20 to 25,000 genes is right answer for the question. Question number 2. Which of the following is correct finding from the Human Genome Project about the composition of human genome? Option A. The human genome contains approximately 1 lakh protein coding genes occupying 50% of the total genome. Option B. The entire genome is composed to unique protein coding sequence with no repetitive DNA. Option C. Protein coding sequence exons constitute only about 1.5% of the genome. 99% of DNA sequences are the same in all humans, and more than 50% of the genome consists of repetitive sequence. Option D. There are no repetitive sequence in the human genome, and all 3.3 billion base pairs code for proteins. What is the right answer for this question? So here the options A, B, C, D. Would you guess the right answer for this question? Let's check whether your guess is right or wrong. Option C is right answer for this question. Let's see why and how. Go to detailed explanation for the question. The HGP produced several surprising findings about the human genome. Most strikingly, protein coding sequence exons make up only about 1.5% of the entire 3.3 billion base pairs genome. The vast majority of DNA doesn't code for protein. More than 50% of the genome consists of repetitive sequence, satellite DNA, SINES, LINES, and transferable elements of no known direct coding function, sometimes called a junk DNA, though many have regulatory roles. At the population level, 99.9% of the nucleotide sequences are identical between any two humans. The 0.1% difference accounts for all genetic diversity in our species, including disease susceptibility variations. The number of protein coding genes, 20,000 to 25,000, was far lower than expected and is only slightly more than the 19,000 genes in the nematode worm C. elegans. So this is the correct explanation and rightly said. The option C is right answer for this question. Next, question number 3. DNA fingerprinting is a technique used to identify individuals based on their unique DNA profiles. Who developed this technique and what is the molecular basis of individual uniqueness? It explicits. Option A. Developed by James Watson in 1953 based on differences in the total length of human genome between individuals. Option B. Developed by Carey Mullins in 1983 based on amplifying random segments of DNA using PCR to create a unique pattern. Option C. Developed by Frederick Sanger in 1977 based on sequencing the entire genome of an individual to identify unique mutations. Option D. Developed by Alex Jeffries in 1984 based on the variation in the number of tandem repeats, VNTRS famously known as, of specific short DNA sequences at multiple loci across the genome. What is the right answer for this question? Would you guess? A, B, C, D. What is the right answer? The right answer of this question is option C. Let's see why and how. DNA fingerprinting, also called DNA profiling, was developed by Alex Jeffries at the University of Leicester, UK in 1984. The technique exploits the fact that certain regions of the human genome contain a tandemly repeated sequence called VNTRS, variable number of tandem repeats. The short sequences 10 to 100 BP repeated in succession at specific chromosomal locations. The number of times these sequences are repeated varies enormously between individuals, except identical twins. So the total length of the VNTR region at each locus is unique to every person. By cutting DNA with restriction enzymes, separating fragments by gel electrophoresis, and probing with radioactivity, a fluorescent proves complementary to the repeat sequence. So Southern blotting, a pattern of brand, the DNA fingerprint is revealed. This pattern is unique to each individual and reproducible from any tissue, blood, saliva, hair roots or semen. That's what option D is right answer for this question. Next, move to question number 4. DNA fingerprinting has several important applications. Which of the following correctly lists established applications of DNA fingerprinting? Option A. Forensic identification of criminals from crimes scene samples, paternity, maternity disputes, identification of disaster victims, wildlife conservation, population genetic studies. Option B. DNA fingerprinting is used only in paternity testing and has no application in criminal investigation because it requires too much DNA. Option C. DNA fingerprinting can identify the exact age of suspect and their blood group from a crime scene sample. Option D. DNA fingerprinting is used exclusively to sequence the entire genome of crime suspects at crime scenes in real time. This question directly related to DNA fingerprinting applications in forensic and paternity testing. Would you guess the right answer? Yes. What is the right answer? The right answer of this question is option A. Let's go to detailed explanation. DNA fingerprinting has broad and powerful applications in forensic science. Take samples from crime scenes, even tiny amounts of blood, saliva, semen, or a single hair with its roots can be DNA fingerprinted and compared to a suspect's profile, providing evidence with extremely high certainty. Probability of two unrelated people having the same fingerprint is less than 1 in 10 billion. In paternity and maternity disputes, a child's DNA fingerprint contains a bands inherited from both biological parents, matching with the claimed parent confirmed biological relationship. In mass disasters, victims are identified by matching DNA with relatives. In conservation biology, a parentage and population structure of endangered species are studied like this. In India, DNA fingerprinting services are provided by the Center for DNA Fingerprinting and Diagnostic CDFD in Hyderabad, so which is many times in the news and very famous. Next, move to question number 5. The Human Genome Project was conducted with parallel attention to its ethical, legal, social implications. Which concern is correctly identified as an ELSI issue of the HGP? Option A. Whether DNA replication in sequenced genomes is faster than in unsequenced genomes. Option B. Whether sequencing DNA is technically possible using only manual methods without computers. Option C. Privacy of genetic information, potential genetic discrimination by employers or insurers, a psychological impact of learning about disease, so predispositions, and equitable access to genetic technologies. Option D. Whether the human genome contains more or fewer genes than the genome of other species. What is the right answer? So this question related to HGP ethical, legal, and social implications. The right answer of this question is option C. Let's see why and how. Option C is right answer for this question. The HGP dedicated 3 to 5% of its annual budget to studying ethical, legal, social implications, recognizing that mapping the human genome creates profound questions beyond biology. Privacy concern arise because an individual's genome reveals information about disease, so predisposition, that may be misused by insurance companies to deny coverage or by employers for discrimination, called genetic discrimination. There are psychological concerns knowing one carries a gene for a late onset incurable diseases can cause anxiety and affect life decisions. Genetic information also has implications for family members who share DNA. Questions of equitable access arise. Will genomic medicine benefit all population or only the wealthy? The ELSI program was a model for responsible science and influenced national genetic information protection laws in many countries. That's what option C is right answer. Question number 6. In the classical DNA fingerprinting procedure, which technique is used to detect the VNTR branch and what probe is used? Option A. Northern blotting is used with the protein probes that bind to VNTR sequence in mRNA samples. Option B. PCR alone is sufficient, no blotting is needed and no probe is used in classical DNA fingerprinting. Option C. Western blotting with antibody probes detects with the VNTR encoded proteins which vary between individuals. Option D. Southern blotting is performed, DNA is transferred from gel to nylon membrane and hybridized with a radioactivity and a chemiluminescent probe complementary to the VNTR repeat sequence to visualize the band pattern. So what is the right answer for this question? So in the classical DNA fingerprinting procedure, which technique is used to detect VNTR bands and what probe is used for this? Yes, option D is right answer for this question. Let's see the explanation. In classical DNA fingerprinting, Jeffrey's original method, genomic DNA from the sample is first digested with a restriction enzyme that cuts outside the VNTR regions, generating fragments of variable length depending on the number of repeats. These fragments are separated by the size using agarose gel electrophoresis. The DNA is then transferred from the gel onto a nylon or nitrocellulose membrane. This transfer step is called Southern blotting, so named after Edwin Southern, who invented it. The membrane is then hybridized with a single standard probe, a short DNA sequence complementary to the VNTR repeat unit that is labeled with 32P or chemiluminescent dye. The probe binds only to fragments containing the VNTR sequence. After washing away, unbound, probe and exposing to X-ray film. So autoradiography or a chemiluminescence detector, dark bands appear wherever the probe bound, producing the characteristic ladder like DNA fingerprint pattern. That's what option D is right answer for this question. Next, move to question number 7. The Human Genome Project uses several strategies to sequence and map the human genome. Which of the following correctly describes two key approaches used? Option A. EST approach, sequence random protein fragments. STS physically maps chromosomes by protein gel patterns. Option B. EST approach sequences only the regulatory regions of genes. STS identifies repetitive sequence only. Option C. EST expresses sequence tags, sequencing short cDNA fragments from expressed genes to quickly identify gene coding regions. STS sequence tagged sites, short unique DNA sequences used as physical landmarks on chromosome form mapping. Option D. EST sequences entire mitochondrial DNA. STS sequences all introns in the genome to map non-coding regions. What is the right answer? This question related to HGP techniques used EST and STS approaches. What is the right answer? Either A, B, C or D. Option C is exactly right answer for this question. Let's understand by the explanation. The HGP uses two complementary molecular strategies to map and sequence the genome efficiently. ESTs involved making cDNA from mRNA expressed genes, then sequencing short stitches from each cDNA alone, from each cDNA clone. Because cDNA is derived from mRNA, ESTs represents only the expressed portions of the genome and provided a rapid way to identify and locate active genes before the full genome sequence was complete. STS's sequence tag sites are short, 200 to 500 BP unique DNA sequences that occur at a known single location in the genome and can be detected by PCR. They serve as physical landmarks or milestones on chromosomes. By mapping thousands of STS, researchers created a physical scaffold of known positions on each chromosome that could then be filled with the detailed sequence data from overlapping DNA clones. Let's move to question number 8. So here match the column 1. So we have terms here in left hand side, and the column 2, it's a correct description. The first one, VNTR. Second one, SNP. Third one, STS. Fourth one, satellite DNA. The same way in opposite side. So correct descriptions for this. The first one, option A, variable number of tandem repeats, short DNA sequences repeated in tandem, varying between individuals. B, sequence tagged site, unique short sequence used as chromosomal landmark in genome mapping. Option C, single nucleotide of polymorphism, position in the genome where a single base differs between individuals. Option D, highly repetitive non-coding DNA sequences in tandem clusters near centromeres used in DNA fingerprinting probes. Option A, 1A, 2C, 3B, 4D. Option B, 1D, 2A, 3C, 4B. Option C, 1B, 2D, 3A, 4C. Option D, 1C, 2B, 3D, 4A. What is the right answer? HGP and DNA fingerprinting key terms match to definitions. Would you guess the right answer? Option A is right answer. 1A, 2C, 3B, 4D. Let's understand by the explanation. The first one matches A because variable number of tandem repeats are short DNA motifs, 10 to 100 BP repeated consecutively at specific loci. The number of repeats varies enormously between individuals, making VNT as the molecular basis of individual uniqueness exploited by DNA fingerprinting. The second one, SNPC, single nucleotide polymorphisms are positions in the genome where a single base varies between individuals. Examples, A versus G at one position. The human genome has 10 million SNPs and they are the most common form of genetic variations used in genome-wide association studies for disease research. The next one, sequence tagged with the sites are short unique sequences at non-chromosomal position detectable by PCR used as physical landmarks to anchor and orient overlapping DNA clones during genome mapping. Satellite DNA, the fourth one, D matches rightly here because highly repetitive sequence arranged in large tandem arrays, particularly near centromeres and telomeres, they beyond density of CS, CL gradients differs from bulk DNA, forming satellite bands, and they are the sequence targeted by proofs in DNA fingerprinting. That's what. So option A is rightly matched here. Let's move to question number 9. So here we have assertion A, every band in an individual's DNA fingerprint is either inherited from the biological father or biological mother. No new bands arise de novo in a child that are absent in both parents. Reason R, VNTR loci obey Mendelian inheritance. Each locus has two alleles, one from each parent, and the band pattern of child is the sum of bands inherited from both biological parents, making parentage verification scientifically reliable. So here are the options. A, both A and R are true, but R is not the correct explanation of A. Option B, A is true, but R is false. VNTR bands are randomly generated in each generation and do not follow Mendelian patterns. So here option C, both A and R are true, and R is the correct explanation of A. And finally, option D, A is false. New VNTR bands regularly appear in children that are absent in both parents due to mutation. Would you guess the right answer for this question? It's related to DNA fingerprinting sensitivity and inheritance pattern of VNTR bands. Option C is right answer. Both A and R are true, and R is the correct explanation of A. Let's see the detailed explanation. Assertion A is true because it's currently performed DNA fingerprint. Every band in a child's profile must match a band in either the mother's or the father's profile. This is because VNTR alleles, like all genomic DNA sequences, are passed from parent to offspring according to Mendelian rules of inheritance. Reason R is also true because provides the precise genetic basis. At each VNTR locus, an individual has two alleles, one chromosome from mother, one from father, specifying particular band position. The child's complete set of bands is union of bands inherited from both biological parents. Each child's specific band is traceable to one parent or directly explains the Mendelian inheritance of VNTR alleles, or is exactly why every band in child must come from a parent. A, in a paternity testing, if all child bands match the mother or pituitary father, paternity is confirmed with proximity exceeding 99.99. Rare mutations can occasionally alter a VNTR allele, but are extremely uncommon. That's what option C is right answer. And the final question, the question number 10 of this topic. Arrange the following steps of DNA fingerprinting in correct order. The first one, P, DNA is extracted from the biological sample, blood, hair, root, saliva, etc. Q, DNA is digested with a restriction enzyme, cuts at specific sequence flanking VNTR loci. R, DNA fragments are separated by size using agarose gel electrophoresis. Yes, separated DNA is transferred from the gel to a nylon membrane. Southern blotting. T, membrane is hybridized with radioactivity. So chemiluminescent probe complementary to the VNTR repeat sequence. U, probe bound bands are visualized by autoradiography. The unique band pattern DNA fingerprint is recorded and compared. So here are the options. A, QPRSTU. B, PQRSTU. C, PRQTSEU. Option D, final, PQSRTU. What is the right answer? The right answer, the chronological order which correctly matched here, it is option B, PQRSTU. It's exactly and rightly chronologically matched here. Let's see the explanation. DNA fingerprinting follows a precise 6-step process. The first one, DNA extraction. Genomic DNA is first isolated from the biological sample using cell release, protein removal. So proteinize K. So precipitation. The quality and the quantity of DNA determines the quality of the final result. Next, move to Q, restriction digestions. The extracted DNA is cut with a restriction endonuclease, such as Henify and Mobile, that recognizes sequence flanking with VNTR regions but not within them, generating fragments whose length varies depending on the numbers of repeats. So here, gel electrophoresis, the restriction fragments are loaded into aqueous gel and separated by size under electric current. Smaller fragments migrate further, producing a smear of thousands of bands. Southern blotting, the separated DNA is denatured, made single-stranded, and transferred by capillary action or vacuum from the gel onto a nylon membrane, preserving the spatial arrangements of bands. So finally, probe hybridization, a labeled single-stranded probe complementary to the VNTR core repeat sequence is added. In hybridizes only the bands containing the VNTR sequence. Its visualization after washing away unbound probe. The membrane is exposed to X-ray film or a chemiluminescence detector to reveal the characteristic ladder like DNA fingerprint pattern. That's what. So PQRSTU, it's correctly and rightly orderly matched here. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you, and thank you so much to all.